Ladies and gentlemen, good afternoon, good evening, good morning, wherever you are. And thank you for joining our event today. My name is Roxy Iqbal. I am the Strategic Advisor at Deep Knowledge Group, and I will be your moderator for today's presentations and discussions. Our event this evening is titled AI in Drug Discovery, Biomarker Development and Advanced R&D. It is being hosted by Deep Pharma Intelligence. Today, we'll give you a complete picture of the industry environment in terms of AI usage in drug discovery, clinical research, and other elements of pharmaceutical research and development. As you can see, we have our agenda laid out for you. Please do take a look at today's agenda. The first speaker will be Preetaman Wadhwa with the topic AI Gap in Pharma Drug Development and Commercialization. The next will be Dr. Mark Keel with the presentation, an AI-driven locus-specific database for 35 ALS-associated genes. After that, we'll hear from Dr. Jamila Amimur with the topic, the state of AI in drug discovery and what to look for. Afterwards, we will have a short coffee break. The speaker after the break will be Dr. Emmanuel Lerner, who will talk about the need for AI in peptide drug discovery. And then Dr. Andrew Ha will finish our webinar with a presentation on exploring sequence-based AI-driven drug discovery, bypassing the need for 3D structure. Please do not hesitate to write your questions in the chat. Towards the end of this webinar, we will have a 20-minute panel discussion with your questions to our speakers, allowing you the opportunity to feedback and to comment and to question any of the content that you have heard this evening. Just for your reference, this event is being recorded and it will be posted for everyone who was unable to attend and for those who would like to reference back to it. So I would like to give a very warm welcome to our first keynote speaker, Preetaman Wadhwa, the Chief Marketing Officer at GATC Health. Preetaman comes with 17 years of global marketing, commercial, strategic lead and Amgen experience. She has also got an extensive background in pharmaceutical, hospital and insurance industry. She has experience in therapeutic therapies, including oncology, hematology, immuno-oncology, cardiology, and nephrology. She has focused in patient-centric healthcare and also has a master's in health systems management. And Preetaman, without further ado, the floor is yours.
Hi, everyone. Good evening and good morning, good afternoon. I see people have joined us from all over the globe. It's an absolute delight for me to be part of Deep Pharma Intelligence Conference today, focusing on AI and drug discovery, biomarker development, and advanced R&D. AI gap in pharma is what I want to focus on today, which should highlight for all of us where we have opportunities to make an impact in the industry as being part of the AI industry. As we think about the pharma and the pharma world, I think today more than ever, the industry is under a lot of pressure. So to generate a new drug today takes more time, more cost than ever before. And as we think about these challenges of what pharma is going through when it comes to showing the value, showing the competition, managing the competition, and also being patient-centric, there are significant gaps that exist for us to be very efficient. In that space, the focus is on innovation, and that's where AI fits in. It's the innovation of science and clinical technologies that can help our patients achieve and make a difference in their lives. The AI gap that exists today is the focus of this conversation because the gap is at multidimensional. We see the gap today exists that even though from an AI standpoint, we've made a lot of progress over the last 2 decades, I would say, maybe more, that we've gone from being able to identify some chemical compounds to overlaying physics, but it's really looking at the whole system and the impact of what our drug discovery is going to have on human systems that seems to be lacking. The second dimension I'd like to speak about is what are we offering as being part of the AI industry for drug discovery? Are we offering a component to the overall service plan, or are we offering the end-to-end services? And finally, looking at the productivity. Such a tedious, long, expensive, highly risky process of traditional pharma drug discovery is of the past. AI will revolutionize how this industry evolves and what time and what outputs are provided, because at the end of the day, we have one goal, and that's all of our joint goal is to say, how do we generate better and safer drugs faster? So my name is Preetiman Wadhwa. Thank you, Roxy, for that background overlay. I'm part of the GATC Health team. GATC Health was founded in 2020 as a technology company where we are bringing multiomics data and AI technology to generate drugs faster by creating a human-like environment. Our platform uses data sources from multiple areas, as well as looking at the proprietary AI platform. We are really able to predict upfront the efficacy and safety of drugs in the entire human system, not just the chemistry, not just the physics, and not just in a cell or tissue, but how the human system will respond to that drug across diseases. And that's really the advantage of GATC Health platform, because this has the promise of drastically reducing the cost, the risk, and the time that is taken to develop drugs today. And just as a brief summary, not going too much into our company, but just to highlight, even though there are about 300 AI companies out there, the way we stand out and differentiate is by offering that end-to-end service. We're not only taking the existing database of 90 million compounds and just matching where an affinity is with the target, but our technology starts from identifying and understanding the disease biology, transitioning that to causality with the drug interaction, identifying those drugs that would work, synthesizing them both at a small molecule and biological level. And that is a unique aspect for GATC Health, which can be applied across various therapeutic areas. And the data source, which is a pretty significant issue that we currently are dealing with our industry in AI for drug discovery. We have our own proprietary data because we have a disease detection arm as well, and offer a flexible business model to work with pharma. Let's spend maybe a few minutes just looking at refreshing for all of us where our problems are in the industry. To nobody's surprise, as we can see here, the pharmaceutical research and development process is tedious and long. Today, as we think about what it takes to bring one FDA-approved medicine out, the pharma company could be spending $2.6 billion behind that one drug, whose peak sales is going to be about $500 million on average. This investment is mostly driven by the failure cost of drugs that are being synthesized in the earlier stage, the first 4 to 6 years of drug discovery phase, where thousands, in some cases millions of these compounds are being tested just to find that they don't work. Even within the clinical trial testing, 90% of these clinical trials fail, which have engaged humans and resources at multiple levels, primarily because the efficacy was not there. The safety signals shown in preclinical were not picked up earlier on that came up in the clinical trials phase, as well as the patient selection. It was the wrong patient or the wrong disease that the trial was set in, and the pathway was not well understood. All of this creates risks for the industry, and the risk-benefit profile is not favoring where we need to be in terms of this investment. $200 billion is what we have invested in R&D just in 2020, and fast growing. And do we think any of this is sustainable for our industry? It absolutely is not. And that's where, from an AI standpoint, there is an opportunity. There is an opportunity to help pharma, partner with pharma across all of these different phases of the development process, whether it is discovery and preclinical, looking at precision disease targeting and optimizing the asset, or looking at the clinical trials and every nuance of what is our lead indication, what is our supportive information on the care that's required, the doses, the safety profile, who is the patient ultimately, and validating up there. And also can be expanded in the later stages of commercialization. My experience has mostly been in commercializing drugs across the globe at various stages of the drug development process. And I can give you numerous examples where at each phase of these drug development, there were failures, there were challenges, and that impacted and reshaped our strategy for the industry at multiple times, sometimes completely eliminating therapeutic areas that were legacy for the company. The good news is we've made progress. 4 years ago, in a journal of biomedical journal of scientific and technical research, this publication came out to say, where will AI partner with drug discovery in pharma across the initial phase of development? This included, will AI be able to enhance the identification of the targets? AI enhanced engineering of the molecules and creating those 3D images? Will we be able to screen AI-based screening and faster throughput in silico and in vitro? The optimization, including being able to formulate, and formulations are more critical now than ever before, especially with COVID. We've seen a shift of the industry from more frequent and more intensive formulation drugs compared to the more convenient take-at-home oral drugs. And then finally, on the CMC side as well. This is all just thinking about the preclinical phase. And I'm pleased to say that even though our initial, over the last 2 decades, progress was low, the last 4-5 years, especially with COVID in the last couple years, we've seen a dramatic change in what it takes to go through these steps using AI. And COVID is a classic example where some of these studies have been super fast and we've been able to bring drugs out much faster. That set a threshold, a benchmark for us as an industry across therapeutic areas. And we've also seen in the next slide, as we look at the clinical trials, the clinical trials and how we did clinical trials has changed pretty significantly. There was a time not so long ago, I would say, what was it, 7 years ago, I was working on a drug in melanoma. It was a very sophisticated oncovirus that we were bringing out. And as we did our first launch, we started thinking about which other diseases in oncology exist where we can inject in the tumor, and started our initial evaluations at that time. It took us 7 years to go through phase 1, phase 2, phase 3 trials, only to know last year that the trial failed, and hence there was no life cycle management. That's really not how trials are set up now. The adaptive trial design, the master protocols that are looking at the basket trials and the platform approach of these trials is really making an impact, and AI is helping in us to be able to partner with pharma and able to achieve which disease, which platform, which patient should we set up our unique trials in. The question really becomes, is that enough? Have we achieved the goal? And the answer to that is we are just at the beginning phase of this wave that is swelling. We have a long way to go as we think about drug discovery and clinical trials taking 10 years to actually bring that drug out. And by the way, on that 10 years, it doesn't count how many decades it took in the academia world to create or identify those biomarkers. We have not shrunk those 10 years yet for patient. And that's a promise to the patients, is how will we bring these drugs, better drugs, safer drugs, faster to them? So while these a couple years ago, Deloitte produced this platform to say this is how clinical trials should be done intelligently with AI, we have not made enough of an impact to shrink that 10 years down just yet. The proof is yet out there. And once the proof is shown, that is where broadly pharmaceutical industry will be able to take benefit of what we can offer from an AI when we offer them the end-to-end service. And what do I mean by that? From wearing a pharma executive hat on, we are constantly making trade-offs. We are making choices to prioritize therapeutic areas that we will operate in, markets that we will operate in. We are trying to look at, we have one drug in a therapeutic area. For example, I worked in neuro, and there was one compound that we were able to bring out, but there was no pipeline. So looking at how do we expand that pipeline to sustain that entire therapeutic area related organization. Every single day, I open news, you open news, and we hear about failed and shelved drugs. The clinical trial failed, and the drug failed in phase 2 or phase 3. Extremely expensive, very disappointing, and huge loss for patients and pharma. Those failed and shelved drugs, if upfront were looked at to ensure that they are going to work in that disease in that patient, or can be repurposed now using AI, is a huge value offering that AI industry can bring out. And finally, we look at where innovation is coming from. 70% of innovation in large pharma is coming from smaller companies. They're coming from outside external sources. And even when pharma starts developing products, half of them nearly are in partnership because of the risk. All of this out-licensing, in-licensing, partnership, think about if that can be validated and confirmed prior to making and sealing those deals, because an AI platform was able to tell the pharma company whether that technology, that drug will work. And finally, from a repurposing to ensure upfront, not 7 years later, like I gave you the example in melanoma, but upfront on day one, as we start preparing for melanoma, we are looking at renal cancers, we're looking at head and neck cancers, we're looking at all other types of tumors where we think an injectable oncovirus can really make a difference and help these patients, so that there's a parallel strategy and not a sequential strategy. This is the promise that pharma is seeking from AI companies, and we are equipped and we need to make our technology so sophisticated to deliver on. Because as we think about future, next slide, please. What will be the value of AI market? From one source called Vantage, they say this market is going to be worth 4.2 billion in 2028. I think that's hugely, immensely undervalued. We could be in upward of 20 to 30 billion. We could be upward of 50 billion. We could be upward of 100 billion, because the impact this industry can make on accelerating innovation has not been tapped in fully. And that's the gap, and that's the opportunity. Next slide, please. We've seen mushrooming of AI companies all over the globe, not just US. While there were 240 companies in 2020, there are over 300 companies in 2021 that are focusing on artificial intelligence and its use in drug discovery and partnering with pharma. And interestingly, it's, while US and Canada is holding 2/3 of it, there is a fast growth happening in smaller markets in Asia and other countries that we'll see innovation and partnership there. We've seen partnerships happening in Japan with the local Japanese company. I think this week there was an announcement between Ono and Iptos, where they're going to focus on multiple diseases and pain management as an area for drug discovery. We've seen huge deals happen between Accenture and Sanofi, Merck, and all other large pharma announcing their deals with AI drug discovery. And that is the beginning of what we have to deliver. Because the challenge is, while traditionally we were focusing on looking at filling just a gap of time, our AI gap is multidimensional. We have the preclinical asset identification ability, which is one very specific task. We used machine learning to solve it, and then realized, oops, we don't have the depth of the quality data that is needed. Hence, the AI gap focuses on so much manual work that has been happening. How many potential lead agents are identified? What time it's taking? What cost it's taking? And if that's still the case, we haven't solved the AI opportunity yet, leaving a gap. And to become more tangible about it, just to put it in perspective where we are, where we can go, and we were at a point where it would take us 4 to 6 years to identify, excuse me, 5,000 molecules or so. And sometimes, like I said, it would be millions of assets analyzed by pharma to identify which drug will proceed into clinical trials. When we think about the existing AI companies out there, what they have done is they have shrunk that time to a couple years, and they've optimized that it's not 5,000, it's perhaps 1,000. And in some cases, very recently, we've seen that they've said, okay, we'll have 200 to 300 of those agents identified, which then pharma can evaluate and take it to preclinical testing. GATC Health, for example, because its technology is so unique and is focused on human system versus just the physics and chemistry, is able to do a little bit more. And what they are able to do is identify 1 to 3 compounds in 4 to 6 months, ready for preclinical testing. And that really shrinks the overall time down. And then process of validating the technology across multiple diseases. But as we think about the future, and I'm taking a very big perspective here to say, how will we succeed? How will we deliver our promise for our patients that we are accelerating innovation and bringing better drugs, safer drugs, faster for them? This silo approach that we have today of the 300 companies in AI highlighting their technology, their unique platform, their offerings, partnering individually with pharma, and then bringing drugs to the clinical trial, that's yet not reaching the efficiency we need in the industry. What we envision is in the future, perhaps there will be a drug discovery platform that is shared across all the entire industry. There are algorithms that are unique by company, hence focusing on a particular aspect of it. The drug design, the drug synthesis is unique, and hence using that common platform, we are moving forward into clinical trials. So finally, I'd like to end my talk by highlighting where we see this industry going, where I see this industry going. I would love to hear your thoughts as well. I feel there is a huge opportunity we have to bridge the gap between the proof of concept to production. Over the last couple years, we've seen how drugs have progressed from just the identification and fitting to perhaps now the synthesis, perhaps now from small molecule to biologics. But we still have to see a proof where a drug comes to market that was started by AI and that shows upfront what will the drug do in the human system, and then deliver on that through the clinical trials across multiple diseases, across multiple patient types, and show the efficacy and the safety. I also feel that we've talked about patient centricity, but perhaps AI still has the opportunity to bring human centricity and fill that gap by digitizing medical data, and that ensuring that not only the physiological aspect of humans, but also the behavioral, the psychological, the demographic, the uniqueness of patients can be brought in so that that precision medicine idea can be taken to the next level, with also offering open source AI. All of these will help us fill the AI gap that exists. We'll be able to transform the industry, and I really think what was taking 4 to 6 years should not even take 4 to 6 months. It should take 4 to 6 weeks, perhaps 4 to 6 days to identify what is that right medicine for the disease that will ultimately help us deliver our promise to patients to make a difference. And that's what I believe our hope for the future will be. That's the end of my talk. I'd love to hear if there are any comments or questions. Otherwise, back to you, Roxy.
Wonderful. Thank you so much, Rishma. That was really informative. I know you have to go quite soon, so we do have a quick question, if that's okay, to ask you, which comes from Grigor Zhushnowicz. Apologies if I've mispronounced your name. How many assets do you currently have in the pipeline?
So we have 2 active assets and then another 6. We are working at GATC Health in neurological diseases, oncology. We have diabetes, Alzheimer's, and then our pipeline is quite rich. We are just getting additional data with which we'll expand further. And our first drug is ready in the market for partnership this summer.
Wonderful. Thank you so much. I know we've had several comments and questions. If you do actually have any time, then, by all means, you can answer them directly in the Q&A. I know Gregor's came back to ask a question about if you are generating any revenue as yet, particularly. So if you prefer, you're welcome to answer them direct in the chat, because I know that you do have to shoot off quite soon.
Yeah, and then I'm on LinkedIn as well. Please reach out to me and I'm happy to address your questions there.
Wonderful. Thank you so much for your time. Enjoy the rest of your day, and we look forward to hearing more from you soon in the future.
Thank you. It was a pleasure being here.
Thank you so much. So everyone, you will be able to get some details about Preetam if you go into the chat feature right at the bottom of the screen. We did share earlier Preetam's contact info, including, I believe, her LinkedIn and profile directly relating to her website. I'm sure we would be able to share that again, which is obviously, and also for GATC Health as well, along with their websites. If we can possibly share that again, there you are. Then by all means, you are welcome to get in touch with Preetam with any questions that you have. I hope you wouldn't mind if people got in touch with you directly on LinkedIn. Thank you so much for joining us. I'd like to, now it does give me great pleasure to welcome our next keynote speaker, Dr. Mark Keel, the founder and Chief Scientific Officer of Genominom. Have I got that right, Mark? Genominom.
You nailed it. That was awesome.
Wonderful. Didn't even need to Google it. Mark completed his MD and his PhD and molecular genetic pathology fellowship at the University of Michigan, where his research focused on stem cell biology, genomic profiling of the hematopoietic malignancies. I got that one right?
Blood cancers, yep.
Brilliant, thank you. And also in clinical bioinformatics, I believe. He is the founder and CSO of Genominom, where he supervises the scientific direction of the Genominom suite of AI-driven software tools. Mark, it's wonderful to have you join us, and we are grateful for your time. The floor is yours. I do believe you have a presentation.
Yep, I'll just switch over the slides, and
Super.
while I do, if I can multitask, I just want to thank Preetam for that presentation. That was a fantastic overview of challenges and opportunities in AI for pharma. For the purpose of my talk, if my screen can be seen, Roxy, is it the presentation mode?
I can see, yeah.
I'm going to take a deep dive into one of those specific aspects that Preetam brought up, which is to say genomics. And I'm going to do that by way of example for one particular disease. Again, the example here being genomics as a great use case for how AI can be brought to life in the service of better treating and identifying patients with devastating disease, and ALS being one specific disease, though Genominom works across disease types, including cancers and rare disease. So the substance of my talk today, very brief, is an AI-driven locus-specific database for 35 ALS-associated genes, ALS being a neurodegenerative disease, and a locus-specific database being the variants that cause the disease in any one of those 35 different genes. So a bit of background about Genominom: the value that our company provides to our clients and our users leverages AI to make genomic information actionable. And we do this in the service of better understanding, better diagnosing, and better treating rare diseases and cancer patients. And on the pharmaceutical side, we use that information to create precision medicines targeted at molecular drivers of disease. Since we're talking technology here, this is one of the core technologies in Genominom's capability. We refer to it as genomic language processing, or GLP, which I may refer to later. I call this slide the order from chaos slide, where on the left is the chaos of the evidence in the published clinical and empirical or functional literature. In the middle there is what our GLP capability indexes out of that chaos. the diseases, the clinical phenotypes that the patients present with, the therapies that have been tried in patients or in animal models or in vitro, and the genes and their genetic variants. Importantly, no matter how an author describes any one of those entities. So not only does our GLP index that information from a broad corpus of that reference material, again, clinical and functional, but we also organize that information into what we refer to as genomic associations. How those genes and the variants in those genes associate with disease causation and what particular phenotypes are occasioned by them, and critical to what we're talking about here today, what therapies have been used to treat them or might be used to treat those different diseases. So I mentioned that GLP is one of our core competencies, and that's exemplified here on the left, which is AI-driven indexing of that data to put together that information into informative associations. The computational component that we bring to this challenge of understanding and making actionable this genomic information ensures maximal sensitivity, making sure that we're not missing anything. So every piece of that evidence is automatically indexed and organized into those associations. But the other flavor of our technology that is also AI-driven ensures maximal specificity of that data, which is a critical necessity when you're dealing with high throughput computational screening of information, such as AI techniques often afford, is to ensure that you've got the highest accuracy. And for that purpose, we have internal AI capabilities that allow our expert team of curators to expertly manually review that information to ensure maximal specificity, accuracy, and appropriateness to specific hypotheses for our pharma clients who have a particular drug development thesis, or otherwise to ensure that we're getting the right information that's idiosyncratic to particular diseases or genes. And for that purpose, we have an AI-driven computational queue or software suite that allows us to do that very rapidly and accurately at scale. And one manifestation of that I want to spend the rest of my talk talking about is in the service of understanding a complicated, clinically heterogeneous, genetically challenged disease, neurodegenerative disease called ALS. I won't spend too much time talking about the clinical features of ALS other than to say it's heterogeneous and complicated, but rather I'll focus on the AI capability that we have brought to bear to better understand this disease. It has a genetic component to it. That genetic component has to do with more than 36 different genes, any one of which, when mutated, can lead to disease. The landscape of information that we produce for each of those causative genes is a deep understanding of which genetic variants, genetic changes in those genes, confer disease to those patients. Based on what evidence? All of which is curated to clinical standards to ensure the highest comprehensiveness and accuracy of that comprehensive causative genetic variant landscape for this devastating disease. So I'm showing some statistics there, but importantly, this is computationally organized for comprehensiveness and expertly curated for clinical grade accuracy. And we're able to do this for any given disease or set of diseases. We put that together in the landscape that I referred to, and there's two types of those landscapes that I want to exemplify here. One is the variant landscape, where the clients that we engage with have a need of understanding all of the variants for the purposes of drug discovery, target identification, or patient recruitment and enrollment, and ensuring that the right patients are enrolled in their trials. And I've mentioned before what this comprises, but I'll emphasize that it's every variant in any one of those genes ever published with all of the necessary evidence to support their interpretation and meaningfulness moving forward for drug development or clinical diagnosis and treatment. This is a summary of the data feeds that feed into and comprise our variant landscape. I've mentioned all of the published literature, the full text, the supplemental data, the figures and tables, etc. All of the clinical information and the functional information that comprises the literature component exhaustively indexed through that computational GLP technique that I talked about. Coming along with that is the functional studies, in vitro, in vivo, in animal studies, et cetera, as well as the population frequencies of these variants, which suggests whether they're benign or potentially disease-causing, and then predictive models of pathogenicity or protein damage that might confer increased risk of clinical disease. All of that information put together is what comprises a variant landscape. And importantly, we have internal tools that fully automate the organization, annotation, and aggregation of that data. We have capability to manually, expertly review that information, and then we follow up that manual annotation and understanding of that data through an automated scoring that conforms to clinical grade frameworks. So the example that I want to give you is one of those 36 plus genes causing ALS, which is one of the main genetic drivers. It's the gene called SOD1. Just to give you a reflection of the power of this combined AI computational approach, is a previous background understanding through an engagement was of 70 causative variants in SOD1, whereas when we were asked to understand the full landscape, we increased that understanding by nearly 5-fold. And I'm exemplifying that here in a Venn diagram where the previous understanding may have come from a public database such as ClinVar, which is a collection of clinically submitted variants that were found in patients for this gene. When you look, though, at the data that we have assembled, which is shown in the crescent in green on the left, we have dramatically increased the understanding of those causative variants, which not only helps you better understand the nature of the variants that cause disease, but also increases your likelihood of identifying patients who are amenable to treatment, particularly when you have a precision therapeutic. This is a different way of looking at that same data now, where we're opposing those variants, which are shown here as circles, against the linear structure of the protein. So when you unfurl the protein from start to finish, and then plot out where those variants appear in that protein and cause disease, what you're seeing here now at the top is the reflection of our comprehensive variant landscape compared to the very shallow understanding that's available absent a AI computational technique. A little bit deeper is, I mentioned that we understand the diseases, the phenotypes, the therapies for those patients. We also understand demographic information such as their age at onset, the disease severity, et cetera, and do so at a very granular variant level. And what you're seeing here is, again, that same protein structure with the variants here now in the different columns, and the circles being reflective of individual or groups of patients who have those particular variants, and on the y-axis, what their age at onset was. So if I focus your attention on the main message here is, some of these variants have a dramatically earlier age at onset for the development of ALS symptoms compared to others. And that's an important indicator from this computational data that we've expertly curated to say that not all variants in this gene are created equal, and some confer exaggerated risk to the patient, in this case in the form of earlier age at onset. I mentioned we also capture information that has to do with the rate of progression. So do you have an indolent course of disease that proceeds over 10 years? Or do you have a very rapid course of disease that takes place over a year or two before ultimate demise? And what you're seeing here is that some of the causative variants, based on the evidence in multiple patients who have that variant, confer a slow rate of progression of disease compared to others that confer a fast rate of progression. So again, this is just an example of the power of AI to organize this data, the power of AI to allow us to curate that information to maximal effect, and some of the types of insight that we can glean from that work. Here's a different way of looking at that data, now adding 2 additional genes, the FUS gene and the TBK1 gene, which number among the 3 dozen that cause ALS, where you see here now the age at onset is plotted along the x-axis, and the number of patients that have those different ages of onset are plotted on the y-axis in these distribution curves for each gene. So SOD1 on average confers a median age at onset of 45, whereas patients who have ALS because of FUS mutations, have a mean age at onset that's a decade earlier, and TBK1 variant patients with ALS have a presentation that's 5-6 years later. So you're starting to see a distribution in the nature of this disease, depending on which of the genes that drive disease are to blame. Lastly here, I'll exemplify our capability again to understand this information at a very deep level, computationally indexing for maximal sensitivity and completeness, and then expertly curating that information with our AI curation capability. This comes in the form of our patient landscapes, where now, before I talked about every variant ever described in any one of these genes, now I'm talking about literally every patient ever described in our entire corpus of clinical and empirical scientific literature. We know each of those patients, we know their genotypes, we know their ethnicities, their ages, other demographic and phenotypic information, as well as their response to therapy. This just diagrams those capabilities that we have of deeply understanding all of these patients and all of their particular presentations. Again, emphasize that this is computationally organized for scale and then expertly reviewed for accuracy, with all of that information, every scientific assertion being reviewed by a human curator. The example that I want to conclude with is now every patient with ALS who has a genetic lesion in the C9orf72 gene, the most frequently mutated gene causing ALS. We have put together a comprehensive patient landscape of more than 2,300 of these patients with all of those annotations that I talked about earlier, which culminates in a deep understanding of things like the nature of their clinical diagnosis and the probability of survival, which is displayed here in these different Kaplan-Meier survival curves, depending on the precise nature of their clinical diagnosis, whether it's ALS or motor neuron disease or frontotemporal dementia, say. Again, all at the hand of any one of those genes that I described. Lastly, here is a distribution of the patients and their ethnicities based on the number of reported cases among those 2,300 plus individuals that we've exhaustively curated for all of those different components that I had talked about. So again, I think Pritiman did a fantastic job of setting this up, where I wanted to take a very focused view on one aspect of AI, and that is in the service of better understanding genetics and genomics, and how that is used to understand the heterogeneity of disease causation and provide insight for pharma into how better to treat that, that genetic disease. So with that, I'll turn it back over to Roxy, unless there are any questions.
Wonderful. Thank you so much, Mark. We do have several questions in the Q&A box, but I think we might save some of them towards the end, if that's okay with you. Wonderful. Thank you so much for such an enlightening presentation. I can see several comments and chats already that are engaging with a lot of what you spoke about, for sure. So yes, I'm absolutely delighted to welcome our next keynote speaker, Dr. Jamila Amimar. Dr. Jamila Amimar founded Mind Centers Global to help businesses and organizations apply AI. She is ranked amongst the top 10 global thought leaders in AI and emerging technologies, and also in predictive analytics. Dr. Jamila is passionate about making AI accessible for everyone. I'm very much interested in hearing what you've got to say about this. So yes, over to you, and looking forward to hearing what you've got to say and your presentation.
So thanks for this opportunity. Wanted to talk today, to take a step back and talk about the state of AI in drug discovery. Roxy, thanks for the introduction. So my name is Jamila Amimar. I'm the CEO and founder of Mind Centers Global, with the mission to help businesses apply AI and investors optimize their AI portfolio. So I wanted first to say, let's ask a question, what is AI? Because AI means different things to different people. So for some people, AI means robots. For other people means, any smart technology, like, if you have like an iPhone or, an Apple Pad, then, somehow you are using AI. For others, using drones is AI. For others, algorithms and, predicting calls and predicting risk and predicting things to happen, that's AI. But also, I wanted just to show the picture in the middle of the faces. So those faces are basically fake pictures. So these are pictures of people that do not exist in reality. It has been produced by AI. So AI means a lot of things. I want to, the definition that I like the most is the one that was put by John McCarthy in 1956. He was the first person to coin the term AI, and he defined AI as the science and engineering of making intelligent machines. And in a way, AI is not just one field, it's a combination of a lot of fields coming together. We are talking about mathematics and statistics and neuroscience and psychology and computer vision, and all those things coming together to form the field of AI. So within AI, you have robotics, autonomous or driverless car, virtual agent. But the most famous, subfield of AI is machine learning. And then within that, we have the famous deep learning that a lot of people have heard of, the progress we had so far around, the computing power. So because now we have so much powerful computer, then we can now execute those algorithms, such as deep learning. For the ones who have followed, for example, DeepMind, the famous AlphaGo, game that has beaten basically a human expert at the Chinese Go game, have used a combination of deep learning and reinforced learning to be able to achieve that. So when we talk about the business, most of the time when we talk about AI in business, we are really talking about machine learning in business. So when we look at the drug discovery area, so how AI has been used so far. So there have been 4 main areas where AI has been helping. One is around drug screening. So this is more about using AI for virtual screening, using AI to identify the targets. We also see AI being used in chemical synthesis. So for example, finding the synthetic routes, predicting the pathways of those syntheses. Another area is drug repurposing. So using AI to identify new targets, or using AI to predict, drug protein binding, for example. Another area is polypharmacology. So this is more about, for example, predicting side effects and so on. So there have been several areas where AI has been used in drug discovery. Now, if we look in terms of investments and funding, so you can see that in 2021, we have funded more than 4 billion in AI in drug discovery. And that amount is the double amount of the money invested in 2020. So there have been a lot of interest, in AI in this area, in particular, after COVID, there have been a lot of money poured into this area. When you look at AI, there are a lot of techniques within machine learning and deep learning that are used. However, there are 2 methods here that I want to highlight. One is the NLP, the natural language processing. So this is all about trying to understand the human language and try, to use it to read research paper and try to, summarize what the research paper, is about and the findings. It's another area is knowledge graph, which is a lot, used a lot in drug discovery in AI. And this is all about, trying to find the relationship between entities and try to find, relationship between different factors. And it's used a lot in drug discovery. Now, when we look a little bit further into in terms of the market share and the application, so the application so far, you can see, you know, pie chart. So most of the AI in drug discovery application, they're around oncology, infectious disease, neurological disorders. Those are, you know, taking the biggest share of AI in drug discovery application. In terms of the projected market share, so we project that the AI in drug discovery market share by 2028 will be around 7 billion. You can see that there are like two regions that are really taking the most portions of that share. The first one is Asia Pacific, followed by North America. Then you get Europe and the rest of the world. Now, if we look at the competitive landscape, and by the way, this is not an you know, an exhaustive picture. I just wanted to take a snapshot and, you know, basically talk about some of those you know, competitors in this area. But if we want to group them, basically we have three groups. We have the pharmaceutical groups, you know, companies. So those are traditional pharmaceutical companies that produce you know, medicine and drugs. And, you know, they have looked into using an AI and investing in AI. Then we have the second group, which are the tech companies. So those are, you know, have nothing to do with, you know, the area of healthcare. They have the technologies, so the likes of IBM and Google and the Microsoft. And then you have the third category, which are the AI startups. So those are the AI startups that are using both the AI, you know, knowledge and using kind of the healthcare knowledge and putting together those startups to, you know, solve, you know, problems within the drug discovery area. So there are examples, for example, where we have AstraZeneca is using, for example, AI to try to predict what molecule to produce next, and is putting money internally. So they're, you know, they're heavily investing into creating their own internal researcher, data scientists, but they're also investing commercially, also in you know, externally. You have the like of Novartis, for example, that has formed a partnership with Microsoft to you know, develop an AI platform that can suggest, for example, molecules. We have also the example of Johnson & Johnson that is using AI in the robotics, you know, area of AI. So they're using AI in what we refer to, you know, basically surgical instruments. When we look more detail into companies, we have the like of obviously Google with its original you know, DeepMind AlphaFold. They have created an algorithm or, you know, an application that can predict protein structure and protein folding structure. They have also announced that they're going to launch a brand new company that will only work and focus their work on AI in drug discovery. We have also the like of, we mentioned Microsoft's partnership with Novartis, but we also have the like of NVIDIA that through its GPU, they have what they call the NVIDIA Clara Discovery. So that's an AI platform that accelerate the computations that are needed to discover new molecules and new drugs. Then you have the third group, which is the AI startup. So obviously we have the famous UK example, you know, Benevolent AI, which is using AI and more specifically, you know, the graph knowledge type of AI methods to try to research literature and try to find connections, to find like new pathways to new components, new drugs. You have the example of here, let's take the example of AI focuses on pathology to try to find what is the drug effects of those different you know, drugs on pathologies. So the key message in here is, you know, you have those three groups competing together, but at the same time, they're also collaborating to progress AI in drug discovery. Now, if we look in terms, okay, so if we look in the future, what are the key drivers that will propel and accelerate AI in drug discovery? There are like two key factors. One is around, which I've hinted a little bit at the previous slide, which is collaboration and partnerships. They're going to be really key, looking forward in terms of AI in drug discovery. Here we have a few examples on how those collaboration are working right now. So we have the example of here new probe with the C2 AI genomics are basically putting together an initiative to screen and monitor cancer. We have Deep Cell and Stanford University that are doing like a project around trying to have a deeper look at around 2 million cells, have a better knowledge and a deeper understanding of cells within our bodies. We have Genomian and Elexon AstraZeneca rare disease that are applying AI to try to have a more accurate diagnosis of rare disease. And we have also the example of AstraZeneca and Benevolent AI that are putting together, that combined effort to try to find, discover new treatments for chronic kidney disease and idiopathic pulmonary fibrosis. So that gives you a flavor on how those, parties are getting together to progress the field. It's not yet proven, but why are all those parties are putting efforts in AI? Because it's believed that AI will reduce the time to discover a treatment, but it also reduce the cost to discover new treatment. And there are like some initial studies that suggest that AI potentially reduce costs by up to 70%. And I guess like the future will tell us how far we are from that estimate. But this is why people are really keen, in applying in AI in this area. Now, there are not, in addition to the drivers and the opportunities, there are also challenges. So one of the obvious challenges is around data. With any kind of tool or technology, if you have like, we all know the old saying that says basically garbage in, garbage out. So if you have poor quality data, you are going to get like poor quality outcome. So one of the challenges is how can we improve volume and the quality of data in drug discovery, make sure that the outcomes of those AI models are good. Another challenge for AI in drug discovery is around interpretability. So how can we interpret and how can we explain the outcomes of those AI models? Some of those AI models are so big and so complex, it becomes hardly difficult to know what is happening inside of the models, especially if you're using, the kind of deep learning. So kind of, companies in this area really need to some time, to improve the transparency and the explainability of their AI models, especially because we all know that healthcare is heavily regulated. So we know, to have like AI models that are transparent and that could be explained. Another area is a lack or the limitation of AI talents or AI capabilities within those companies. So there is a lot of efforts that needs to be addressed and together to build those internal data scientist team, internal AI research researchers teams. Companies need to invest on those capabilities for AI to be successfully applied in this area. Another area that I wanted to highlight, but I'm not going to go into much details, but it's kind of the technical aspect of AI in drug discovery. So we know, for example, when looking at a drug, for example, or a new treatment, it's not about, they're like, you have several objectives that you would like to optimize. It's not just one. You would like to find, like, for example, a drug that is efficient, has the lowest number of side effects, that can go with maybe with other drugs. So you will have several functions that you try to optimize, and that could be an issue when you apply very simplistic AI models. So you need really to have and develop a little bit more sophisticated AI algorithms, AI models to cope with this multi-objective of optimization within an AI context. Now look at, so what are the elements to make AI successful for drug discovery projects? So we talked about alliances, collaboration, and partnership. We mentioned the AI talents and capabilities. We also mentioned data, the technology ecosystem that needs to go about, because you need to have a good volume of data and a good quality of data, but you also need to have infrastructure that can facilitate, the use of that data, the transition of data from one phase to another. But another two components that are really important and sometimes that get missed, along the process. One is an AI strategy. You really have to have a robust AI strategy in place to be successful. So you need to know where you are now, and what is your vision, or what is your ambition, then determine what will be the building blocks to get you there. If they're like, and I'm going to exaggerate, but if there are going to be 100 ways on how you can apply AI, and we have seen there is just one way to apply AI, there could be several ways on how AI could be used in drug discovery. How can we identify the ones that we the most value to the business? And all that, it has to be done, under that AI strategy. Then the other component is business integration. So whatever strategy you come up with, whatever AI platform you end up developing and using, it has to be integrating within your current business processes and the procedures. Otherwise, if you don't do that, you will end up, basically putting your product on the shelf. You need to have a business integration that shows how AI is being used on these companies, that shows the governance, structure around that, who has the accountability, who signs on that, AI outcomes, and that's kind of a way on how you integrate your AI model and, your AI platforms in your current process. So I think this is really what I want to show, a flavor of what we can look for, in terms of the drivers, the challenges, but also some kind of like tips and guidance on how you may have, successful projects. Happy to take questions afterwards.
Thank you so much. That was really enlightening. We do have, I can see quite a few comments in the chat and some questions, but I think let's go through them towards the end during the panel session, if that's okay with you. I would love to introduce you. So next, we are going to hear from Dr. Emmanuel Lerner, the CEO and co-founder of Pepticom. Dr. Lerner is a scientist and biotech entrepreneur with a proven track record in envisioning and establishing AI-biotech startups and projects. He co-invented, founded, and is leading Pepticom, a disruptive company in the field of AI-based peptide drug discovery and design. Dr. Lerner led Pepticom from concept to revenue generation via collaboration with large multinational companies, negotiated and executed discovery deals, and also seed and round A funding for finance. Dr. Lerner is also a biochemist with 15-year academic prior to the experience academic prior to the establishment of Pepticom, and includes a PhD in biochemistry, followed by postdoctoral research at the Hebrew University of Jerusalem. During his studies, he published research in leading journals such as Blood, Nature and Communications, the Journal of Clinical Investigation, Cancer Research, and others as well, and had the honor of publishing his postdoc research with the 2017 Nobel Laureate in Physiology Medicine with Professor Michael Roshbash. That is quite impressive. Thank you so much, Dr. Emmanuel, and over to you. Looking forward to hearing what you've got to say.
Thank you, Roxy, for the introduction and the ability to speak in this very interesting event. And the rest of the former speakers were really excellent and a very good introduction to right into what I'm going to say. So Pepticom is into really the discovery and optimization of peptide-based drugs. And I will talk about the need for incorporation of AI into the drug discovery and development in peptides specifically. So you may know that we are celebrating the 100th year of insulin, which saved millions of lives. And it's basically a large peptide. The peptide is basically a short protein built from a string of amino acids. And as you may know, most of the peptide drugs on the market are either hormones or derivative of hormones. But in the last 10 years or so, we are starting to see completely synthetic peptides getting into the market as potential drugs. And this is changing the peptide market dramatically. It is very challenging to bring this a completely novel peptide because you have to have a way of discovering these peptides. In a very short, I would say that the peptide drug market is projected to double in the next 5 years to something around $51 billion due to these new medicines. So thinking about discovering a new drug and you don't have a hormone to start with or a native peptide to start with, you need a screening method or a discovery method. So a breakthrough was done and the Nobel Prize was given to phage display, which is basically building a huge library, physical library of 10 to the power of 9 peptides. I won't get into too much details, and basically throwing them on target and getting an active peptide, and then with very hard work, with a lot of time and effort and cost, optimizing it to a drug. And after this breakthrough came another breakthrough, which was the invention of basically what is called mRNA display technologies, which is an elaborate way of doing something like a phage display with 10 to the power 13 molecules without a phage, which is a very impressive technology. And this also enables the incorporation of non-natural amino acids, which is very crucial in many cases in peptide drug discovery to make the peptides more drug-like. So this brings us to the current stage of where biotech and pharmaceuticals are as far as discovery. And what you can see here on the screen is a successful discovery project. And as you heard before, discovery and development of drugs in general and peptides is still a very inefficient process. 85 cents lost to each dollar put into R&D, and around $2 billion of drug development and discovery if you take into account all the failures. So this is a very good story. This is a peptide that is in phase 2 from Merck MSD, which is an orally available, fully synthetic peptide. So this is not a derivative of any known peptide in our body that was discovered using a display technology. In this case, something that came out of Rough Pharmaceutical, collaborating with Merck MSD. And what you can see here, and that the reason I'm putting this, first of all, this is going to really drive the peptide market, but the time that it took to discover, from discovery to an early hit was 3 years and $4.5 million, and then another 5 years and $12 million just for the chemical optimization and discovery of the molecule. This doesn't take into account of all the other costs around. And when you think about that, that's really a very, very long time. And there really is a need to try to make this process more efficient. So why is it so difficult to discover peptides using these methods, which are really amazing methods as far as the biochemistry? So there are a few very basic things. First of all, you are talking about a chemical space which is around 10 to the power 30 different solutions. And these techniques are screening only 10 to the power 13 solutions at best. So they're not screening not even a millions or billions of the real problem. And the second thing is, once you have an active peptide, because this is a chemical library, you have no idea where this thing is actually binding. So it's very difficult to optimize these molecules to drug candidates. So you spend years in trying to understand how the structure activity relationship basically in these cases. And this is really time consuming and very, very cost ineffective. And the third thing is the fact that when you're using a library, you don't really control almost anything. I mean, you put the natural building blocks of peptides, you may incorporate a few non-natural amino acids, and you know what the size of the peptide is, but you don't really control any other aspect. So you can have a very good binder which has a horrible solubility. This was briefly discussed by one of the speakers. And all of the above are things that, at least in concept, can be addressed by artificial intelligence. So what we want to do and what the direction the world is heading to is basically moving from hormones to what we have today, which is library-based new drugs on the market, to drugs that are basically designed via artificial intelligence. This would enable, as we heard, and more effective time management, reduction in cost and risk, basically. So the way we believe in Pepticon and the way forward in our case of new peptide drugs is using reinforcement learning. And we are doing this because of many different reasons. One of them is the kind of data that is available today. Taking into account that data on cyclic peptides, and we are talking about small cyclic peptides, you can't really build on big data here because there isn't a lot of information or data around these kinds of molecules. So we choose reinforcement learning. Basically what we do is we take a structure. It can be a crystal structure, it can be, if there is a good prediction from AlphaFold, it can be a prediction. We use our algorithms that were developed in Pepticon, and then we basically do an in silico discovery process where we have different strategies trying to find the optimized molecule. And once the run is over, we synthesize the peptides and we actually test them in-house and validate them and then optimize them. When we started a few years back, our first publication, that was an academic collaboration with Oxford University and Hadassah Hospital in Israel. We had a very primitive version of the technology. We had only natural amino acids and linear peptides. We got very nice results. I won't get into the paper, but we found a very interesting peptide that could tackle an ultra-orphan disease. But we understood very rapidly that this is not good enough in order to compete in the peptide drug field, where you need really superior attributes for your molecules. So we started working on cyclic peptides and heavily non-natural modifications of these peptides in order to give them the right qualities they need. And we got to a point today where we have a very, very diverse chemistry, when we can control basically a lot of aspects of the physiochemical aspects of the peptides that we discovered in our AI software, basically, which is a very good situation to be in. And just as an example, you can see here in red the 20 natural amino acids, natural building blocks, but there are hundreds of other building blocks that most of them can be used in drugs that are, in most cases, very hard to incorporate into screening methodologies and we have basically programmed 300 different building blocks. So we have a much more diverse chemistry basically to play with when we try to discover these new potential molecules. Another aspect, obviously very important, is the ability to predict the outcome. And we are working on that as well, but I won't get into too much details here. So as I showed you before, we are working with chemistries of cyclic peptides, non-naturally modified. This enables very active peptides, and I will show a few examples in a minute. One of the outcomes of these chemistries is very stable peptides. You can see this bar graph where our peptides are extremely stable in human serum. One of the known problems of peptides is their poor stability, which is a very bad starting point as far as a drug. So this is just an example. We have many other examples. We run our software on AWS. It's basically an ephemeral supercluster that can run parallel projects in the same time. And here is an example of a project. We focus most of our activity on in-house projects, but we have also collaboration with large companies. So this is a project where we started a very, very challenging protein-protein interaction inhibition on an autoimmune disease where there's a real need for new therapies. And we started in the left. This is an inhibition test with a hit molecule. And you can see that as we go to the right side, the potency of the molecule increases. So these are iterations on the molecule. And then we were able to reach a lead potencies as far as the biochemistry. And this is a very good indication to the fact that our model is correct, because if you have a wrong model, you'll never be able to optimize a molecule unless you synthesize thousands of molecules. And we did, you can see here, some other tests, and this is now tested in mice in this auto-inflammatory model, and we got very, very nice results that basically are in par with standard of care. And we have other interesting aspects of this molecule, but I won't get into more details here. This is another example where we took us 3 months to discover a peptide, which is basically an inhibitor of the SARS-CoV-2 interaction with the human receptor and basically cell entrance. Bear in mind, these molecules are completely synthetic, so they don't look anything like a short version of one of the proteins involved. These are completely synthetic, novel peptides. Our aim is basically to improve the technology and bring it to a stage where we need to screen less than 100 peptides in order to start from scratch, from an idea to a very highly active molecule. Obviously, there'll probably be a need to play a bit more with the chemistry, but really bring a valuable molecule that can be used in the pharmaceutical or in the biotech industry as a very promising starting point, because it's very difficult to bring like a hit which is not extremely potent and you don't know how it works. So you need this validation, and this is exactly why we do a lot of work in-house. And basically the company itself is a mixture of computational chemists, software engineers, and biochemists trying to work together and look at the project as a whole from a holistic point of view, because we believe that there's really the need to connect the dots and bring something, in order to bring something that has value in the industry. So this is why we work in such a way. It's very challenging, obviously, to have such a multidisciplinary approach. And we are very fortunate to have two really key persons, key opinion leaders in the industry, Dr. Walid Danho and Dr. Tommy Sawyer. Both of them actually discovered and developed drugs that are in the market today. So they give us a lot of insight about how to do this in a correct way. And that's it. Thank you very much for your time.
Thank you very much, Dr. Lerner. That was really interesting. And I think there are a few points that I want to touch upon, but we'll do that during the panel session, if that's okay. And again, please do go onto the chat and have a look for Dr. Lerner's contact information, his LinkedIn, and also details for PeptiCom as well. It would be great for you to engage directly with his posts, and I can see that you are actively promoting this conference as well, so thank you very much for that as well. So without further ado, I'd like to introduce to everybody our final keynote speaker for this evening, which is Dr. Andrew Ha. Dr. Andrew is the Senior Manager of Deogen. He has a PhD from the University of Stanford. He strives to advance Deogen into the international market by accelerating global business plans and spearheading collaborative partnerships. Dr. Andrew, it's a pleasure to have you with us today. Feel free to, provide a little bit more in depth of an intro for yourself. I was curious to see what you had your PhD in, and absolutely, it would be lovely to hear your comments on today's session.
Yeah, thank you so much for the introduction. Thank you so much. I did my PhD in stem cell biology at Stanford. I was also a scientist at Genix Therapeutic, which studies immunotherapies. And after that, I joined Deogen, which I'm going to talk about today. Let me, all right, can you guys see the screen?
Yeah.
Oh, perfect. All right, so I'll be introducing our AI platform, which utilizes the protein amino acid sequence to identify novel drug candidates. And this approach opens up a great potential for AI-driven drug discovery and development by bypassing the need for 3D structure. All right, so today's talk is divided into 3 sections. The first 2 sections are devoted to presenting our sequence-based drug discovery platform. Next, I will introduce our company structure. So I want to start off this talk by giving you one example of how Deogen's platform identified remdesivir as a potential drug for COVID-19. Remdesivir was approved by the FDA as the first treatment for COVID-19 in July 2020. However, Deogen was able to predict and publish a paper in January 2020, even before the 3D structure of SARS-CoV-2 was identified. And how do we predict this drug candidate without even knowing the structural information? The answer is we made this discovery by taking an unconventional approach. So instead of relying on structural-based approach, which is commonly used by most AI companies in the drug development space, we use the amino acid sequence of the target protein to predict drug candidates more quickly and efficiently. And why is that important? Approximately only 8% of total human proteins has been crystallized, and drug targets from AI-biotech companies that take a structural-based approach are restricted to this 8%. In contrast, the amino acid sequence of most proteins are already known. This means that 92% of human proteins that could serve as potential targets are overlooked. So our hope is that by utilizing the protein primary sequence, we can tackle this unexplored 92% in the drug discovery space and increase our chances of identifying novel drug candidates. Deogen's innovative technology has been recognized by a top global report, and Deogen has been selected as one of the top 33 leading AI companies in drug development for 2 consecutive years. Unlike most AI companies in this space, we have an end-to-end platform that supports not only the initial stages of a study, such as from target discovery to hit identification, but also lead optimization and preclinical development. And all of these advantages derive from our core AI technology shown here. For example, we integrated transformer model into our platform, which is used primarily in the field of natural language processing, but we are the first to apply it to the field of drug discovery. In addition, our platform includes a wide range of deep learning and machine learning algorithms, and these methods are used for target selection and drug candidate design. In addition to these advancements, we also have additional components that are unique to Deergen's platform, which support the prediction of binding affinity, binding site, and determines the prediction of uncertainty. I would like to briefly go over our three major platforms. First is Deer Trends, which is used for target discovery. We have developed a neural network-based feature selection algorithm called WX. Once we input publicly or privately available transcriptomics, such as NGS or microarray data, our platform can rank genes based on their discriminative index score, and which then can be used to select an optimal set of genes for target or biomarker discovery. Next, Deer DTI is our drug target interaction platform that is used to identify hit molecules. Once we input protein and compound information, our platform can predict and provide binding affinity scores, as well as efficacy scores that is used for hit discovery. What I'd like to reiterate about our drug target interaction platform is that it is a sequence-based approach. On the left shows the type of input that we utilize. For small molecules, we use SMILES, which represents chemical compounds in the 1D string format. For proteins of interest, we use the amino acid sequence, which is also a 1D string format. Most AI companies requiring the 3D structure as the input are limited to candidates with a tertiary structure. However, Deergen's sequence-based approach bypasses this limitation. The right side shows the type of output that can be generated. Deergen's platform provides the binding site and binding affinity, as well as functional activity scores such as IC50 and EC50. Lastly, MolEQ is our lead optimization platform. Once we have a seed molecule, we can optimize the molecule until a desired threshold is achieved through a reinforcement learning process. In addition, our platform predicts both a final compound and the reaction pathway based on available chemical building blocks. Our platform is unique in that it can optimize substructures of the molecule while keeping certain part of the molecule the same. And what's important to note is that our model considers multiple properties at once and optimize the compound with differentially weighted parameters of interest, which is critical for tailoring the best compound for specific needs. Now here is an overview of our operating system with a flow chart of each of the platforms that I just described to maximize our AI learning capability in the drug development process. We have recently also established our own R&D center so that we can actively pursue targets identified in-house. And by performing wet lab validations on our identified compounds, we generate more data that can be fed back into our platform. And this creates a closed loop learning system that allows data from experiments to fuel future design. And this continuous reinforcement loop drives the perpetual growth in the power of our predictive models. I'd like to share with you two case studies. In the first example, we are targeting Alzheimer's disease. We use our drug target interaction platform, which uses target amino acid sequence. And in this case, FDA approved drug libraries as inputs. Within 2 weeks, we were able to identify hits, and our experimental validation of the predictions looked very promising. Here, you can see the predicted and experimental EC50 values look very similar. The second case study shows one of our oncology projects. Here we have identified novel molecules for PROTAC or proteolysis targeting chimera, which utilizes targeted protein degradation as a mechanism for cancer therapy. We were again able to identify hit molecules in 2 weeks, and the results show a very potent anti-cancer effect. So for the last part of my talk, I would like to briefly give you an overview of our company. From our founding in 2016 until 2022, we have shown consistent progress and establish effective partnerships with leading pharmaceutical companies and academic institutions globally. Today, we have over 20 projects in collaboration with 10 partners, and we believe that our R&D center will spearhead active expansion of in-house pipelines. We also established a site in the US to strengthen external collaborations. Currently, we have 4 sites, 3 in South Korea and 1 in the US. And to give you some context of our team credentials, we have equal representation in AI technology and life sciences. Currently, we have over 50 full-time employees that are experts in diverse fields. Our leadership board consists of leaders with 20 plus years of experience in AI and biological sciences, as well as business. And lastly, the following table summarizes our co-development and in-house pipeline programs. Our co-development programs are pursuing indications in various fields, including oncology, NASH, and CNS. Our in-house pipeline is currently in development, and we look forward to sharing details of our progress in the coming years. And that's all I have for today. With that, thank you for listening, and I'll be happy to take any questions.
Thank you so much, Dr. Ha, for your presentation. It was very, very informative. I think we have certainly got quite a few comments and questions relating to what you've discussed. So I think with that, we will now like to start our panel discussion. We have got a remarkable group of panelists with us. And I think what we'll do is we have a set of questions, obviously, that have been thrown out there by quite a few of our attendees. And so what I would encourage our participants to do is please do think about what you've heard. And if you have any comments, then now is the time to certainly ask your questions, and I'll try and get through them as quickly as I can. So I think let's first of all ask all of our speakers present, if you wouldn't mind kindly taking yourselves off mute and also coming back online so we can see you. And obviously, if you aren't speaking, then do feel free to put yourselves back onto mute as well. So let's, I'm going to start with some of the more general questions that we've had. One from Edward Hudgens, who says, hello from Washington, DC. Today, he is revising FDA proposed reform legislation. And if you could reform or completely redo FDA to allow for innovation of discovery and certification process, what exactly would you actually challenge and what would you do? So I'm going to throw that out there for one of you to try and answer and maybe have a think about. And, you know, it would be good to have one of you feedback onto that as well. If you could
think that it will take a
something.
lot of time to actually be able to show the ability to, you know, to prove to the FDA that you can actually dramatically shortcut or some of the processes. So we've seen that obviously with the COVID, but bear in mind that the technologies themselves were, you know, technology that were worked on for sometimes a decade. So there wasn't like a real dramatic shortcut, and this was like a very, very specific emergency. So I think that once there's enough evidence to show that you can actually have, let's say, an excellent prediction for, let's say, toxicity or something like that, and this is shown for a few years, maybe there's a possibility to have a discussion with the FDA, but that's, you know, it's a very tough thing to do. That's my take on it.
Would anybody else like to comment on that?
I was going to tell you that you were muted, but I'll pick up where Dr. Lerner left off and say that there's the primacy of evidence, even when you're talking about AI. And so the approach that we take is in mining the evidence and presenting that evidence. Approaches that have been touched on from other speakers here might be to produce more evidence. And so you'd hate to, especially in clinical circumstances, solely rely on predictions in the absence of that evidence. So it's about finding more of that evidence and refining it and qualifying it. And that's the only way I think you're going to see any change in the FDA's requirements.
Going back to you, Dr. Mark, there was another quick question that we had, which wanted to specifically look at the genome query language. How is AI used with that?
Yeah, sure. We have a different approach to AI where we're not using black box techniques. We have more of a heuristic bottom-up approach. And so undoubtedly, panelists and attendees are familiar with the IBM Watson and their approach to solving this challenge of pulling out genetic and genomic information. We feel like the flaw in that logic was to have enough data to have the patterns just self-assemble and emerge. And we took a different approach, which was to say, understand the myriad different ways that genetic variants can be described and all of the different ways that genes are discussed, and build on that early success and iterate from those early successes, as opposed to taking all of the data and asking agnostically for the patterns to emerge. We found the simplest ways to codify those patterns and built on our early successes, and that's what's culminated in our very comprehensive approach at this point.
Lovely, thank you. Dr. Ha, quick question for yourselves. How long does DHN take to analyze a sequence?
Yeah, so that's a good question. So it really depends on each project. But for example, on average, to identify hit molecules, we can do it as fast as 2 weeks. But it could take a couple more weeks, depends on the availability of the data sets and how well known it is. So yeah, on average, 2 weeks.
And a general question to throw out there as well that was asked earlier is, what about the technological challenges that you might be encountering? It's great to hear about the success stories, but to understand for a lot of people as well who are attending, they may be encountering some of these. It would be good to hear from yourselves about the challenges in terms of the roadblocks with technology and what you have done to overcome them.
I can refer to a few challenges. So first of all, very interesting comment from Andrew. But I have a question to Andrew. So
yeah, sure.
2 weeks. So you're basically selecting a molecule from a known chemical library, right? Or
that's correct. We can. So what we can do initially is to use publicly available data sets, but we have collaboration that a collaborator brings privately available data set that we can also analyze. That also helps to identify novel molecules.
Mm-hmm. Very interesting. So as far as challenges, first of all, we use, as Andrew mentioned in his talk, we do need the 3D modeling or a good structure, and this is still, even though there was the DeepMind breakthrough, people tend to think that this solved all the problems. It didn't. So you can have, in some cases, an excellent model, which is a computational model, and you can have a horrible model. And it's also, it depends on the site on the protein that you're trying to tackle. So there's a lot to do in this respect. That's one challenge as far as we are concerned. Luckily for your company, that's not an issue. And the other issues are around synthesis. So we are fortunate that with peptide synthesis, it's quite, straightforward. So it's a string, basically. You can have some, to play some games with it, like a cyclic peptide with something, a lasso peptide, things like that. But the synthesis is quite straightforward. In many other cases, that's not the case, and you can have anything from a very easy to straightforward synthesis to something that is almost impossible to synthesize. So that's another issue that you sometimes tackle when you try to find an innovative molecule.
Yeah, so I guess I can also add on to maybe one of the challenges that we face. I think one of the biggest challenges is obtaining data that are qualitatively meaningful. So as we all know, technology opens the door to greater data accessibility, but selectively identifying right set of data for a particular project is critical.
Yeah, I agree with that, Andrew. I think data is really a big issue in terms of the quantity and the quality of it. And I think this is where we see, collaborations and partnerships forming. So you can see, pharmaceutical companies forming, for example, partnership with universities who do more research and collect more data, or, partnership with another commercial organization to, try to sort out, the data issue. Because without it, you cannot then, train the model and then, gain that accuracy you need on the outcomes.
Lovely. Actually, Dr. Jamila, touching upon that, I'd be interested to understand what challenges are inspiring you. Obviously, AI promises to improve lives through science and innovation. So with these challenges, what specifically is inspiring you?
AI potential, I'm really passionate about AI, and I think, the potential is significant. Obviously, there are, the good things and the things about AI. I see AI as a tool. So it's the way we use it. If we use it like in a good way, then it becomes good. If we use it like in a bad way or for a bad, objective, then it's bad. So I think, the potential is there, whether in business, whether saving lives. We've seen the theme of AI for good really, expanding over the last few years. We've seen AI, being used to improve, or diagnose and improve mental health. We've seen AI being used to, help with deforestation, for us. We've seen AI, try to predict earthquakes. We've seen AI in healthcare. There are like so many, outcomes, that is like, good for the overall humanity. Then, you really need to be aware of some of the issues around AI. Obviously, we haven't talked about a lot in here, but like a big issue with AI, apart from the data, is the bias. Again, it's not necessarily AI or the algorithm that is biased, but it's the data that is fed into that could bias. Maybe it's the way, the person who built, or construct that algorithm that could have a biased opinion. So if we've seen, for example, some really example where it went really, really bad, in terms of bias. So for example, Microsoft first Twitter bot, become very racist within 24 hours of interaction with people. We've seen the Amazon recruitment tool that, favored men over women for the same, for technical roles with the same kind of CVs. We've seen it in the US, justice and so on, and also kind of like in some healthcare application. So I think the good, the potential is that it's just like, the way you go about it, you need to be aware of all these. So you include all those things in the design phase, because if you miss the design phase, it becomes very expensive to correct it, when you are, towards the end.
Absolutely. I mean, I was actually going to ask in terms of, are AI applications always successful? I mean, there will be failures, and perhaps other panelists want to contribute towards this as well, is what, how can we avoid when we are working with AI applications and they just simply don't work?
So maybe, Roxy, if I can build a little bit, and then I leave it to the other speakers to add. So, as I already mentioned, AI is not always successful. Obviously, it has been successful, especially in healthcare, in terms of, so it's very good with computer vision, for example. So the use of deep learning has increased the accuracy rate for computer vision accuracy by more than 40% compared to the traditional, methods. So we have some, success example. We have some, failure examples. I mentioned, the bias, in the, Amazon, Microsoft, bot tool. But there is another famous, I think it's called, it doesn't have a name, but like the way, you describe it is the image basically of a husky. So there is a husky with the snow on the background. Then you ask AI, is it a dog or is it a wolf? And then it wrongly classify a husky as being a wolf. But then what they found out is basically why there was an error, because AI was not really looking at the image of the husky, it was the snow. Because there was the snow, it has automatically, kind of detected that it must be a wolf. And I know, it must, it's just like, kind of like an animal type of, kind of example. But the same thing, can happen with, in healthcare. So some of the application that came up, with reading CT scans for COVID-19, the chest scans. So some of the studies find out that sometimes AI has wrongly identified, for example, a patient, having COVID or not by reading the scan. And some of it, was to do with the position of the CT scan. So for example, if the CT scan was taken with the person, standing up, or if the person was laying down. So for the people, for the images where the people were laying down, AI, has given, a high likelihood of, severe COVID. Which you think, there is a sense, because if they were lying down, it means they were potentially not well enough even to stand up. But, you can see, it has detected that pattern, and then it has made the conclusion that any CT scan where the person is laying down, it means that it is severe. Where we know in reality, it's not always the case. So you have to be at, kind of like, aware of those kind of biases, those kind of like, wrong correlation that could, happen in practice. I think because of what we do, and then maybe, the others, speakers can contribute. I think first is to be aware, because if you are not aware of it, you cannot, sort it. So the first one is awareness and education around the, AI capabilities. The second one is, as I said, the quickest way to sort out, things is during the design phase. So during the design phase, for example, you need to have like some, ethical principles, some procedure on how to check for bias, maybe some procedure on how, you go and look a little bit in terms of the data and try to understand it and so on. But I think, the design phase is very, very, very critical.
Naturally, absolutely. Would anybody else like to comment upon that as well?
Yes, I would. Very good examples from Dr. Jamila for computer vision. When I'm looking into drug discovery, so I think two things. First of all, it's a very broad question, but two things that I think are important to take into account. First of all, you're doing a simulation of some kind of reality. So computational power will only increase, and the cost would decrease. So even if you reach a failure, you'll probably reach it much sooner and much cheaper than using conventional methods. So even if you take a very grim scenario of a failed project, you'll probably be in a much better situation compared to years of spent research on a wet lab as far as drug discovery. So that's my take on the project, on the question.
Dr. Mark, Dr. Howard, was there anything that you would like to add?
Well, just to summarize, my thinking there is, if you have a failed proposition, is you might reconsider your question, and a different slant there is re-examine the tools that you've applied to seek out an answer. And so if you're continually meeting with failure, you might ask a question with some more precision and increment your way toward the larger answer, as opposed to asking higher level questions that may be challenged by the specificity intrinsic in your data set.
Absolutely, absolutely. Dr. Ha.
Well, I think everyone covered what I was going to say, so yeah.
No, I think it's a very good point from all of you, Dr. Mark, to actually review the idiosyncrasies of, particularly when you're looking at AI in a grand scale. As you've said, Dr. Jamila, you have to really think about, first of all, understanding, and then actually looking at the design phase and breaking it down block by block. And it's not as simple as just kind of assessing it as a wide question to throw out there about failures in applications, but really, as you were saying, Dr. Mark. It's highly dependent on each case and each actual design that you're trying to implement, I suppose. As opposed to looking at failures in application. So thank you so much for making that point. We have run over, and I apologize. And I just want to say a really big thank you to all of our speakers. I've been really inspired by some of the conversations, by your work, by the contributions that you are making in drug discovery as well. So thank you so much for that. And of course, if I could ask that in the chat for us to finally add the links to all of our speakers and their profiles and where they work. So I would invite our participants and all of our attendees today to review a little bit further the works and the experiences of each and every one of our wonderful and acclaimed speakers today. Thank you very much to all of you. So with that, I would also encourage everybody to who actually attended today to subscribe further to our events and our updates. You can see the link at the bottom, which is www.deep-pharma.tech. And stay tuned for additional reports, analytical reports, and IT platform releases and announcements that we will have, and updates on the subject of advanced technologies in the pharmaceutical industry. And so please do check out for our latest reports. We have one coming up on artificial intelligence for drug discovery, the landscape overview for this quarter. So I would also just like to quickly let you all know, Deep Pharma Intelligence is building a very sophisticated cloud-based engine for advanced market and business intelligence in pharmaceutical and healthcare industries. It includes a data mining engine, infrastructure for expert data curation, and advanced visualization of dashboards, including mind maps, knowledge graphs, and a three-dimensional visualization of the ecosystem. The analytics can be delivered to fund targets to investors and to companies, and also contain detailed SWOT analysis as well. The platform is a powerful tool for investment institutions, so I would encourage everyone to definitely keep informed about that. And also for private and public companies in the pharma and biotech space, research institutions, universities, and more. Essentially, it provides investment analytics and actionable insights about primary and secondary markets, SWOT analysis of the most promising entities and technologies, providing a clear view of opportunities and risks, identification of growth opportunities, including partnerships, technology deals, and transaction prospects, competitive intelligence, and business and technical due diligence, and more. So it's quite exciting that we will be releasing this quite soon, and I do believe it will be a very valuable tool for everyone attending today to certainly keep informed about. Okay, with that in mind, thank you so much to everyone for attending once again, and for joining today's presentations. As I said, that there is a recording of this that will be shared for all of you to be able to reflect back on and to use as a reference point as well. Thank you so much, and I very much look forward to seeing you all at future events. Thank you so much, and good evening.